Welcome to the introduction of genetics. So today we're going to go over a couple topics that are really rudimentary to the study of genetics. The first one being the history. So, many people might think that genetics is a rather modern field of science, but that is not quite true. So the first proof of genetic theories being used was 15,000 BCE around. And this is when we first noticed an attempt at genetic engineering through artificial selection, which is the deliberate breeding of plants and animals for desired characteristics. One example of this is corn. So regular corn, not artificially selected, has pretty small cobs with not very much and pretty small kernels. Now, through artificial selection of only breeding the biggest plants, we now have our modern day corn, which is pretty large and has nice plush kernels. So how people have figured out that artificial selection was used during this time was analysis of pollen in hermetically sealed ancient tombs, and from the dogs also buried in the tombs. So now we're going to jump a little bit more into the future, to the 5th century before the Common Era, and we get people like Hippocrates and Aristotle. So Hippocrates mentioned that hereditary material consists of physical material. There's something there that heredity is based on. However, at the time, he didn't quite know what it is. He just kind of was like, oh, it's there. Now, Aristotle, he had another idea. It was that people can transmit characteristics that they do not show at conception, but develop at a later age. So his observation, once again, was imperative to the understanding of genetics, but was a little bit before we fully understood it. Now we're going to jump pretty far in the future to the 17th and 18th century, and we get notable scientists like Van Leeuwenhoek, who made the microscope, so we are finally able to see things with smaller than what the naked eye could. We had people like Linnaeus, Lamarck, Lyell, Darwin, who came up with natural selection. And then by the time we get to 19th and 20th century, we get people like Wallace, Galton, Mendel, who is termed the father of genetics, and we'll be learning more about Mendel later on in the series. And then we get into modern biology, modern genetics, which is what you think of today. So, before our understanding of genetics, what did people believe? How did they think we got new things? And they believed at the time in spontaneous generation, which is new cells coming from old cells, like new stuff coming from old stuff. So, for instance, one of the theories was that maggots came from meat, because people often noticed that on meat there were maggots. Also, like ants came from bread, was kind of like another idea. However, and this was disproved in 1668 by a scientist by the name of Redi, and he did this through a maggot study. So he had 3 containers, and he filled each container with meat. The first container he left open, and a couple days later, or a little bit later, when he came back, he saw that there were flies and maggots on the meat. Now, in the cork-sealed container, there was no formation of maggots in the meat. And then he also had a gauze-covered container, and with that one, he did see some maggots on top of the gauze. However, once again, there were no maggots on the meat. So therefore, maggots, the fly larvae, doesn't come from the meat, because in the latter 2 experiments, there were no maggots on the meat. So therefore, the maggots came from something else. We now know it to be fly eggs, but at the time, they didn't. Now, another experiment was made a little bit later, a couple centuries later, in 1862, by Pasteur. So he wanted to look at cells, bacterial cells. So what he did is he also had, 3 kind of experiments going on. So he had a fancy flask with a neck, and so for the first experiment, he applied heat to it to kill any bacteria, any organisms in it. He let the flask sit, and then later on, he noticed there was no bacteria present. He did the same thing with applying the heat, but then he snapped off the neck and let it sit. And he noticed that there was bacteria present. And then he did another experiment where he once again applied heat, but this time he tilted the flask sideways, and let it sit for a little bit, and then when he turned it right side up, he saw bacteria was present. Now, since the first one, there was no bacteria present in the red media or liquid, so therefore the bacteria itself did not come from the media, but rather from the air, or like the surroundings. Because in the two experiments that the flask was open to the surrounding air, there's bacteria present, but in the one it wasn't because of the long neck.